added. I did put for module 3, I did put test your knowledge, but it's really more about the structures, right? The cell, their function, whether prokaryotic cells have it, eukaryotic cells have it. And then the last one has one for plants too, although we don't focus on plants necessarily for this class, because we don't have anything that falls into microorganisms when we look at the plants. All right, so in our 4th module, there is a reading that goes along with this, and I go to the review. Make sure you go to the review and you notice it, because if I don't have a question in regards to it in the review, this was even in the when we had a hard copy chapter. We're not going to cover, there's a section of the chapter that talks about propagation of viruses, and we're not going to talk about that in this class, so you're not going to get tested on it, okay? And viroids, okay, are also there, and I'm not going to do the viroids. I do the viruses, both the bacteriophage and the animal viruses, and I do the prions as far as the discussion and topics, okay? And then there is also a second little reading that is going to cover just a little brief discussion, which we'll talk about class briefly too, is the role that viruses play in some cancers, okay? So that's what's going on for this particular module. All right, so for module 4, what we've got down is our 4 learning objectives. So identifying the properties of viruses, which we've already looked at the properties of the cellular organisms. So we're going to be kind of looking at those acellular organisms. Recognize the structure and function of the components of a viral particle. Identify characteristics used to classify viruses. Differentiate between the different replication cycles, the lytic cycle and the lysogenic cycle of the bacteriophages. Recognize the steps in replication of the animal viruses, and then kind of distinguish the differences between the two. And then recognize the role viruses play in cancer development. We will also cover the prions. We have already talked about it in the first module, and then we're going to look at them a little closer to this. So, key properties of viruses. First of all, viruses are acellular. So there's no cytoplasmic membrane. There's no cytosol. There's no organelles. There aren't ribosomes. So many of those characteristics that you guys just got quizzed on of what all cells have, these guys don't have. Okay.
Well, I thought they were acellular, so that means they're not cellular.
Correct. That's what I'm getting for. Yes. Okay. So these are not cellular organisms. They are not considered living organisms. They are referred to as infectious agents. They are intracellular parasites. They have 2 stages of their existence. That which is extracellular, when they are outside of the host cell, and that stage where they're intracellular, when they're inside the host cell. Those 2 states are very different. The virus, depending upon the virus, its genome can be either DNA or RNA, never both. If it is DNA, it might be double-stranded or single-stranded. If it is RNA, it could be single-stranded or double-stranded. Okay, so it doesn't always match up to what we see in the cellular organism. The virus cannot carry out any metabolic pathway on its own. The smallest the smallest genome for a virus carries about 4 genes, and that's it. The rest of the genetic information, or the rest of the proteins it needs, the instructions, or it doesn't carry those instructions with to make these proteins, because it doesn't make those proteins. What it does is it hijacks the host cell's machinery. So it hijacks those proteins that the host cell already has and makes those proteins perform the job it needs to do in order to multiply within that host cell. A virus does not grow in size. It does not respond to the environment. It cannot independently reproduce. The viruses can infect many different types of cells. So it would depend upon the particular virus that you're looking at as to what type of cells it infects. So we have viruses that infect, of course, we're all aware of the ones that infect us, because at some point in our life, every one of us has had a viral infection at one point or other in their life, right? They infect animals, they infect plants, and there are even viruses that infect bacteria. The specialized viruses that I mentioned that we were not going to cover in this class, the viroids, in that chapter, they actually infect fungal cells. So you see across the board, viruses are not limited to just, animal cells. They can infect many different types of cells. Now, a virus that infects an animal cell is not going to infect bacterial cells. Okay, it's a different virus. It's going to infect these different organisms. But basically, all types of organisms are susceptible. The viruses that infect a bacterial cell are referred to as a bacteriophage, or just phage. Okay, and you're going to see that as we move through this. We're going to be talking about the bacteriophage replication process as we move through this chapter. All right. Most viruses infect only one particular host cell, and that is due to that interaction between the surface proteins on the virus with interacting with surface proteins on the host cell. Okay, there's a direct interaction, protein-protein interaction, that allows for specificity to ensure that that virus is going to infect the right host cell. Okay. There are viruses that can cross different species and even different genera. Okay, so there are some viruses that infect, say, the dogs that we also can get. Okay, but then there are others that are very specific to, say, the canine group versus with us. The viruses that can infect many different kinds of cells are referred to as generalists. Now, when we talk about viruses as far as size is concerned, that's what's showing here, relative sizes. When we talk about viruses, generally we're looking in the nanometer stage, or unit. Nanometer would be 10 to the negative. Anybody know? 9, 10 to the negative 9, okay? So when you are talking about cells, you're usually using microns to the negative 6, right? Viruses are usually 10 to 100 times smaller than that. Now, the largest of the viruses are on the same size range as the very smallest of the bacteria. Okay, so it is possible that they be a bit larger in size, but in general, we're usually, we're usually using that unit of nanometers when measuring them out. All right, recognizing the structure and function of the components of the viral particles. So we've got the genetic material. As I've already mentioned, the genetic material is going to be either DNA or RNA, never both. If it is DNA, it can be double-stranded or single-stranded. If it is RNA, it can be double-stranded or single-stranded. Now, when it comes to single-stranded RNA, You also have what we call positive sense and negative sense. Positive sense single-stranded RNA is like messenger RNA. When that genetic information enters into the cell, it can be immediately translated, just like messenger RNA. Negative sense single-stranded RNA is like a complementary copy to messenger RNA. When that genetic information enters into the cell, it cannot be translated. The first thing that has to happen is they have to, the virus is going to get the cell to make a complementary copy of that. Now, we don't have the proteins that allow for us to take RNA and make a complementary copy of RNA. Our cells don't do that, which means that particular genetic information and the protein that does that particular activity must be carried with the virus, okay? So, and brought in by the virus. So with the genetic material, it is possible that some proteins will be brought in also, usually limited on the number of proteins. The capsid is the containment for that genetic information that protects the genetic information while the virus is outside of the host cell. The capsid provides that protection for whatever nucleic acid that the organism has. It is also where any proteins that it must carry in with it is going to be stored. The capsid is composed of proteins, and those proteins are referred to as capsomeres. The capsomeres may be of a single type or multiple types. Okay, and we will in a little bit talk about the actual structure of the virus and how different structures that it can be presented as. So when we look at a virus, all viruses are going to have the capsid and the genetic material. Okay, those two combined, you can also oftentimes hear termed as the nucleocapsid. So basically, the two words combined. Those two features all viruses have. Some viruses, though, will also have an envelope. An envelope is a phospholipid bilayer that surrounds the nucleocapsid. That envelope is acquired from the host cell's cell membrane. So the phospholipids are originating from the host cell membrane, and we will talk about how it acquires it as we go into our animal viruses, okay, and how they replicate. Envelope viruses that envelope then will display proteins that allow for the virus to interact with its next host cell. Those proteins are referred to as the spikes. Okay. So if the virus has an envelope, the means for interaction with the next host cell is going to be on that envelope through the spikes. If the virus does not have an envelope, then the means to interact with the next host cell is going to be one of those capsomere proteins as part of that capsid. Okay, so whatever is on the furthest outside of that cell is where we're talking about. So when you look at these two pictures here, right? This is without an envelope, this one has an envelope, okay? And you see, if they don't have an envelope, this is referred to as a naked virus. If they do, it's referred to as an envelope virus, okay? The naked virus, it is just the nucleocapsid, genetic information with the capsid, protein capsid structure, and you notice part of that capsid structure is going to be those proteins that are going to interact with the next host to recognize the next host. The enveloped protein is one where the nucleocapsid is inside, the envelope is on the outside surrounding, and then our spikes are within that envelope. Okay, so attached to that envelope. A complete, whether it's an enveloped or naked protein, a complete protein structure ready to infect the next host cell is referred to as a viron. Now, oh wait, now before I get to this one, let me go back to this one just for a moment. This picture here is demonstrating, so these two are examples of animal viruses. This one is demonstrating a bacteriophage. Okay, the bacteriophage has the nucleocapsid as well as other components. It's what we're going to be referring to as a complex structure. Okay, but it too though has that capsid and genetic information as part of the structure to then move while outside of the host cell. Once the cell, the virus moves into the host cell, the capsid is and the envelope, are, if it has it, are going to be removed. Okay, so it's only the genetic information and any specialized proteins needed for reproduction that will enter the host cell. Now, as far as the structure is concerned, we have 3 different types of viral shapes. And part of the classification process will include separating by these different shapes. We have the 3 basic shapes are the helical, polyhedral, okay, and the complex. So the helical is as what you see right here on the left, and so the red is the genome, okay, so that genetic material. And basically in this case, we're seeing a capsomere of the exact same thing that then wraps around, kind of like when you guys talked about tubulin in your general biology with the cytoskeleton, right? It's one protein unit that then links together to create like a slinky type of, or straw type of effect, where then the new genetic information is inside, okay, protected. This can be with or without an envelope. This is showing without an envelope, okay? But it could have an envelope. The polyhedral is oftentimes an icosahedral. An icosahedral is a 20-sided structure, okay? So the exact same shape, 20 times, fit together to give you this sort of polyhedral structure, okay? And then the complex, and again, this can be with or without an envelope. So these are just showing the nucleocapsids. And then the complex is one in which it doesn't necessarily fall into either or, okay, of those 2 categories. So it's a little bit different and is a little more involved in its structural makeup. The figure I showed on the last one with that bacteriophage, again, that would be in addition to an example of a complex structure. All right, now how do we classify these viruses? Excuse me, not microorganisms. The viruses are generally classed on the type of nucleic acid. So what is their genome composed of? DNA or RNA, and single-stranded, double-stranded, what, right? Whether it has an envelope or not. The shape, the size, sometimes even by the particular host, right? When we are looking at classification and the viruses, we don't go any higher than the families. So there's no kingdoms, no divisions, no classes. Family is the highest level. All right. So now we're going to go and start talking about the actual replication process. And we're going to start with the bacteriophage first. Okay, the bacteriophage can go through two different types of replication processes. They can go through what we call a lytic cycle, or they can go through a lysogenic cycle. The lytic cycle is one in where, that particular cycle is one in which, that the virus is going to lyse the cell at the end. That's the name lytic cycle. At the end, the cell will have broken open, lysed open, releasing those virons. So the lytic replication is one in which there's basically 5 stages to it. We have attachment, penetration or entry into the cell, biosynthesis or synthesis of the genetic information, the proteins, right? Maturation or assembly, and then of course the release when the cell is lysed. So, in this case, we're looking at a bacteriophage. What's unique about bacteriophages is that the bacteriophage is always a DNA genome. Okay, so there's always a double-stranded DNA genome. In addition, the bacteriophage itself, the capsid never enters the cell. Okay, the only thing that enters the cell is the DNA, so the genome of the bacteriophage. So the first step, of course, would be attachment. So in this case, we've got, these are referred to as the tail fibers of the bacteriophage structure. The tail fibers are what's going to interact with the surface of the cell, identifying this as that interaction is specific, identifying that it is a bacterial cell. Okay, because this particular viron is only going to infect bacterial cells. So it is going to attach to the surface. In the structure, it looks like a little alien ship, right? It has a sheath, a tail that has a sheath over it. It has a base of the tail. It has a little tail. They're called tail pins, and then of course we have the head where the capsid structure is and the nucleocapsid is. Okay, when interaction occurs and we have attachment, this structure kind of sits on to the surface, and from the base, those tail pins, we have release of proteins, which are going to digest or break down that cell wall. And then the bacteriophage it's kind of an injection process. It injects the genetic information into, then once it's created an opening across that cell wall, it injects then that DNA into the host cell. Okay, so into that bacteria. So now the bacterial cell has the bacteriophage genome inside. Okay. Once that happens, the genome of the bacteriophage is going to its presence is going to hijack the machinery so the proteins within those cells to redirect them to produce then multiple copies of that genome. So copies and copies and copies of that genome, as well as all of the proteins necessary to produce the capsid. Okay, now, this picture is somewhat misleading. Those proteins to produce the capsid are not going to all of a sudden be in the correct shape, okay? There's parts and pieces, and it is during the maturation step where it all gets assembled into that structure, okay, into that viron structure, the final structure. So during the biosynthesis, we're getting all the proteins being made, the DNA being made. In that process, of course, the host DNA is going to be broken down because those nucleotides are going to be needed as subunits to make viral genome. Once the cell has produced that DNA and the proteins necessary that moves into the maturation phase. The maturation phase is where that capsid assembles. So all the parts and pieces for the capsid assemble together, and they package the genome into the capsid head of that bacteriophage. Digestive enzymes then will cause the lysis of the cell. Okay, and so the cell lyses and the bacteriophage are then released into the environment. Yes?
Is it just me, or does that remind you of the movie Alien? Like, is it just, it's all I think of right now.
Could vary. I won't lie. I'm not a sci-fi fan. As much as I like science, I'm not a sci-fi fan. So I never watched that one. I never watched most of those, but yes.
So is there any of the host DNA left, or none at all?
No. It breaks down.
Oh wow.
It breaks it down into, it's going to use those nucleotides. Those nucleotides are going to be used as subunits to create all this, the viral. Now, a single cell can produce hundreds, like they're just showing a few here, but when this cell lyses, and if you actually see an electron microscope picture, there could be hundreds being released all at the same time. So that genetic information, it needs the subunits, so it's going to break it down. That being said, in this chapter, side note, I forgot, and thank you for bringing it up because it reminded me, there is a conversation about transduction in this chapter, okay? We're saving that for the next unit when we talk about transferring genetic information, because it is we'll discuss in detail later, but if you already started reading this chapter, you might have come across the transduction information, and it really just has to do with packaging accidentally. Sometimes little bits of DNA from the bacterial genome will accidentally get packaged instead of viral genome. And that is a way in which bacterial cells can have genetic information transferred from one cell to another cell. Okay, but you'll notice I'm not talking about it today in detail, or this week in detail, because it won't be on this chapter test, okay? As far as this unit, we'll be discussing it in detail in the next unit, all right? This is kind of one of those downsides of having, a textbook that is slightly organized differently than previous, texts. So just to give you a heads up, focus on the material on that review. If the review's in Unit 1, if the review's discussing it, then yes, it's going to be on the test. If there's nothing at all in the review about it, then it's not going to be there, okay? All right. Now, for the lytic cycle, when we are, we are observing the, bacteriophage present in our sample, right? What we're going to see is a numbers situation that looks along this lines. We have a culture of, say, E. coli cells. We'll just throw that out there, okay? Then we have a culture of E. coli cells in a test tube, and we introduce some bacteriophage. Initially, what we're going to find is that if we were to take a little bit of the media, not the cells, just the media, and we were to then find out how many bacteriophage are, free in that media, what we're going to find is the numbers are going to drop pretty quickly from what we first added in a short period of time. Is going to drop down to pretty much nothing, because the bacteriophage will be inside of the host cell. There'll be none that are free in the media. As time passes, right, and then because this is a lytic cycle, all of a sudden when these cells burst open, we're going to see a very quick jump in the number of these virons in the environment, so in that media, because of the fact that the cells are releasing them all at the same time, right? And then we are then going to see kind of a pause there for a bit, right? And maybe see a drop off, depending upon depending upon our culture of E. coli cells. We might even see a dip in the number for a little bit as they enter in the next group of cells, right? And then the cycle occurs, cells burst open, we're going to see again another jump. Okay, at some point it will remain pretty stable, just because our cells themselves, and for these bacteriophage to enter, have died off. Right, so eventually we'll reach kind of that plateau effect. All right. Now, the lysogenic cycle. The lysogenic cycle here, this is going to be a slightly different situation that's going to occur. This is kind of a modified replication cycle. In the lysogenic cycle, once the virus inserts its genome into the host cell, that genome is then going to insert into the chromosomes of that host cell. And it is going to then be replicated with this cell. So there's no lysis of the cell. There's no production of virons to be released into the environment. The virus is increasing in numbers as the culture increases in numbers, right? So every time the cell divides, that cell is going to, now we have not only one cell going to two cells, but we now have two viruses. Each of those cells has the genetic information for that virus. When those two cells replicate, now we have a total of 4 cells. All 4 have that genetic information from the virus. Okay, and that continues. So they refer to that as a modified replication cycle, because in this case, the goal is not to lyse the cell. The goal is to tuck away and kind of hide and continue to propagate with the cells themselves. Okay. During this time, there is no production of proteins for the viron. So the capsid proteins are not being made. It's just the virus genetic information that's being maintained within the host cell's genome. So the infected host cell then grows and reproduces, and in that process, the genetic information for that bacteriophage also increases in numbers, related to the number of cells. Now, what benefit does this serve? When the nutrient are, and the nutrients are low and the population is not replicating, it's kind of in a stagnant situation, then sometimes it's the ideal then for the virus genome to just hold within those cells, just exist in those cells and replicate slowly with those cells. When the population is growing, the population of cells, bacterial cells, is growing proficiently, okay, that's when then, in order to increase the numbers, it plays to the benefit of the bacteriophage to get out of the cell and then, make many get out of the cell by lysing it and infect all these other cells. It'll increase its population that much faster. Okay? Now, there's only a certain group of bacteriophages that have this capability. They are referred to as the temperate phages. And those, what they do is when they infect the cell, so when the first steps are pretty much the same. Okay, we have interaction with the host cell. We have digestion of the cell wall so that the bacteriophage can inject its genome into that host cell. The genome then incorporates into the genome of that host cell, and that is referred to as a prophage. Okay, so this is referred to as a prophage. The prophage is then going to replicate as the cell replicates. Okay. Now, when conditions become necessary, then the cell is going to, or when environmental conditions trigger it, the prophage will leave the genome, so it'll be pulled out of the genome, at which point then we move into the lytic stage, and it'll start to go through biosynthesis, followed by maturation and lysis.
How does our bodies then fight things off?
You what?
How does our body then fight that off?
What body?
It's not in our body, the viruses?
These are bacterial.
That makes more sense, okay. Never mind, sorry.
No, these are all this is the lice. So far, we've only been talking about bacterial viruses, the bacteriophages, and how they infect the bacterial cells. So you'll notice in these figures, there's no compartmentalization. That's our next step, is to talk about animal viruses. And that's going to be very much different because animal viruses are going to have to, animal cells are different, they're compartmentalized, right? And so we're going to address it differently. Things are going to be addressed a little bit differently, okay? But to that point, lysogenic cycle, this idea of a prophage, there are some animal viruses that can do this. And they are then called, when they're inserted into our genome, they are referred to as a provirus. They are then tucked away, right, into the genome, and they replicate with our cells. And it makes it more difficult, obviously, for our immune system to address it. Oftentimes, these proviruses can be linked to the cancers, because where they insert into the genome, they disrupt some of the, cell cycle control genes or those oncogenes and stuff like that. So we'll talk about that a little bit. But, and proviruses, unlike prophages, unlike the bacteriophages where they can come into the genome and then exit the genome, the proviruses, those are the animal viruses that do something similar, except that once inserted, they cannot be removed. So they don't, once inserted, they're there. They do not pull back out of the genome. They do not have that capability to remove again. All right. Now, when the prophage is present, this actually can at times provide benefits to the bacteria. It is the incorporation of prophage into a bacterial chromosome is called lysogenic conversion, and during that time, some of these bacteriophages will carry genes that alter the phenotype of that bacteria. So some of our bacteria that cause disease only cause disease when they are infected with a prophage. So the fact that they are infected by that bacteriophage and it is inserted as a prophage is why that bacteria then is able to cause disease in us as the host for the bacteria. Okay? So they can alter the virulence factors for the bacteria to make them more disease-causing for the bacterial host. So that's like 3 levels here. We got the bacteriophage. The bacteria is the host to the bacteriophage, but we are the host to the bacteria, right? So there's 3 different things going on there. And the fact that the bacteria is infected then means that for some bacteria, at that state, then they are able then to cause disease in us. All right. Now, steps in the replication of animal viruses. So, animals, the animal cell is different, right? In the sense that we have compartmentalization. We have the nucleus where the DNA is found. RNA is found, can be found in the nucleus. Yes, because that's where it's made. But in the long run, RNA is then going to be transported into the cytoplasm where we see translation occur, right? So that's going to come into play in regards to how the events occur. The events are going to still be somewhat similar, right? We're still going to have the attachment, so that recognition or interaction with the host cell to say, yes, you know, this is the cell that we infect. But then, and then we're going to have entry. Entry can vary. Unlike the bacteriophage, where the capsid always remains outside of the bacterial cell, for the virus, for an animal virus, the capsid can actually enter into the cell. It doesn't have to, but some do and some don't. Okay, it just depends upon that entrance process. If the capsid enters the animal cell, not bacteria, if the capsid enters the animal cell, then once in the animal cell, we have an extra step that we didn't see with the bacterial process, and that is called uncoding, because the fact that the genetic materials inside the capsid, and so that capsid will have to be removed or uncoded from that genome to release the genome. Right? Because in the end, again, the genome is what's important. So that's what we're trying to get into the cell. And then once that happens, we'll see the replication. So our biogenesis is going to occur. Then we're going to see maturation, and then we're going to see some sort of release. It is not always a lytic process. It depends upon the situation. All right. So we have 2 different things going on here. Unfortunately, they gave us 2 examples. They've got the influenza virus. In this case, the book has provided us HIV virus. Both are actually, though, enveloped viruses. So let me figure out how to bring up the, which is not always the case, right? We have naked viruses that can cause disease. So attachment. Attachment is going to be the interaction between the viral protein on either the envelope, if it's an envelope virus, or on the capsid, if it's a naked virus. And that interaction is going to be to a cell surface protein. So for instance, in this example, they're demonstrating CD4 is that cell surface protein that the spike interacts with, okay? That interaction is what is going to trigger the entry steps. Now, there are different types of entry steps that can exist. We have direct penetration, we have membrane fusion, and we have endocytosis. Okay? So there's 3 different main ways that the virus can enter. And it depends, again, it depends upon the virus as to how it enters. Direct penetration would be when this is something that's seen more with a naked virus, where then the naked virus then basically releases the genome into the animal cell. Okay, capsid remains on the outside. The membrane fusion is going to occur with envelope viruses. In this case, then the envelope is a phospholipid bilayer, and it's going to fuse with the phospholipid bilayer of the animal cell. Okay, and when it does, its contents, which is the nucleocapsid, is going to get inserted into that animal cell. Okay, as such, then we will see uncoating occur, where the capsomeres are going to be removed from the from around the genetic information. Okay, and then the last is endocytosis. So endocytosis would be where, and this can be for either a naked virus or an envelope virus, but that is where then the interaction triggers the cell to engulf and bring in the nucleocapsid into the cell, and uncoating occurs. Okay, any of those events are going to happen. And then once the genetic information is inside the cell, it'll depend upon what type of genetic information as to where the next steps are going to be. If the genetic information is DNA based, then the DNA is going to go into the nucleus, where you are going to see then replication of that, so the biosynthesis is going to occur in the nucleus. If the genetic information is RNA, it'll stay in the cytoplasm. Now, HIV is a unique situation where the genetic information is RNA based, but its replication occurs within the nucleus. Okay, and that is because when this RNA enters into the cell, this is the HIV is what we call a retrovirus. It reverse transcribes the RNA into DNA. Okay, so it has to bring that protein with it when it does so, because of the fact that we don't have an enzyme for reverse transcriptase. We do not have the ability to take RNA molecule and use it as a template to make DNA, right? That is a specialized protein that the retroviruses have that allow them to do so. Once they take that RNA and reverse transcribe it into DNA, the DNA goes into the nucleus where it stays and where biosynthesis occurs. Okay? That's a unique situation. You pretty much all your other RNA viruses outside of your retroviruses are going to stay in the cytoplasm. Okay? And your, as I mentioned, your DNA viruses will stay in the nucleus. All right. Now, strategy for once in the right location, whether it be the nucleus or the cytoplasm, biosynthesis is going to occur in that location. Assembly is going to occur in that location, and then go to release. If it is a naked virus, release is going to be through lysis, and it will lyse the cell and release all the naked viruses. Okay. If it is an enveloped virus, what you're going to find is a budding process, like what you see here. They don't release all at one time through lysis because they need the phospholipid bilayer for the production of that envelope. And you say, well, if they're using the cell membrane's phospholipid bilayer, then isn't that a cell membrane? And the answer is no. Okay, any membrane has phospholipids. It's a phospholipid bilayer. That's what separates then the two bodies of the inside versus the outside, right? What makes different membranes, what makes each membrane unique are the proteins that are inserted, right? So that means the Golgi apparatus has a membrane, but it's not cell membrane. Why not? Well, the enzyme, the proteins inserted in the Golgi apparatus are different than the proteins that are inserted in the cell membrane, right? The actual proteins, and whether they integral or peripheral proteins, are what makes that membrane unique to its particular characteristic, whether it's a cell membrane, nuclear membrane, endoplasmic reticulum, Golgi apparatus, right? So the genetic information, the nucleocapsid, gets in, for instance, for HIV, the nucleocapsid wraps around, or cases the genetic information, which is RNA, and then that nucleocapsid interacts with a portion of the cell membrane that is lined with these matrix proteins. It tells it where to exit. And that area of the cell membrane is no longer cell membrane because the only proteins present are viral proteins. So it's like a little, it's like a little signaling. You exit from here, and when you exit from here, you're gonna have phospholipid bilayer with viral protein only, and therefore that is the envelope. The enveloped viruses are going to have to be released slowly over time, so that the cell can produce more phospholipids, so that we have more material to make that envelope for that virus. Okay, so lysis is naked viruses, and budding process is the means by which we see for the enveloped viruses. Lysis is a quick release of a lot at one time as the cell breaks open, versus budding process means we have a slow, these are oftentimes referred to as persistent infections, right? Because they're just slow release over an extended period of time. All right. I'm debating whether I want to go into all right, so for, so those are the steps. So for the purposes of replication. So if we're looking at double-stranded DNA, that's going to go to the nucleus. We're going to, we already have double-stranded DNA in the nucleus. It's going to replicate just like the genome is going to. The main example of single-stranded DNA happens to be the one in which it's single-stranded when it's in the nucleocapsid, but when it enters in the cell, it folds back on itself to become double-stranded. So it's a single DNA molecule, though, that has double-stranded because it base pairs with itself. Okay? And therefore, in the cell, it functions just like a double-stranded DNA. When it comes to RNA, RNA has multiple options. We have, in this case, I've got an example listed of single-stranded negative sense RNA. So negative sense RNA is the RNA that's complementary to messenger RNA. So when this particular genome enters into the cell, the first thing it has to do is make a complementary copy of that is positive sense. That's the one that can be translated and provide protein. Okay, so it has to do this first so that it can produce viral proteins, right? And to do this, it needs an RNA-dependent RNA polymerase that is provided by the virus. So when this particular type of virus enters into the cell, it's going to have the genetic information plus this protein. If it doesn't have this protein, then the virus can enter, its genetic information can enter in the cell, but our cells would have no idea what to do with it. The ribosomes can't translate it, right? And there's no way to replicate it. So the virus is dead in the water right there. It's not going to be able to reproduce. So it must bring its own viral RNA-dependent RNA polymerase so that it can make a copy that can be then, that becomes the positive sense, which is messenger RNA, like our messenger RNA, and it can produce viral protein. It will also, of course, some of those copies will be used to make copies of the genome, right? Serve as the template to make additional copies of the genome. Now, if it is single-stranded, if we come in with single-stranded positive sense RNA from the virus, then that can be translated immediately, okay, to make proteins, right, viral proteins. That's a B. Okay, one of which is going to be an RNA-dependent RNA polymerase. So I'm going to just put RNA D, RNA polymerase, poly. Okay, so that it can make a single-stranded negative sense RNA, okay, that will be used as a template to make our, so that serves as template to make our single-stranded positive sense RNA. And so with all the base pairing, and this is what gets packaged in the nucleic acid. Yeah. Right? So that's easier to deal with. The RNA, the viral RNA polymerase is necessary because we don't have the means to do this process right here or this process. But the virus doesn't have to bring it with, because this is like messenger RNA. It can make it first in the cell. Once made, then do this step. Okay? And then if it's double-stranded, if it is double-stranded RNA, well, in that case, you've got that double-stranded RNA, right, can separate the single-stranded RNA. Negative sense RNA is going to use the template. To make more genome, right? And the single-stranded positive sense RNA is going to be translated for protein. So that the one that's most challenging is if it is a single-stranded, a negative set single-stranded RNA. Because again, that one they have to bring, the virus has to bring with it the viral protein, okay, in order to be able to replicate. The other two options, single-stranded positive RNA or double-stranded RNA, they don't need to bring the protein with, because one way or the other, they're going to have a messenger RNA, a viral messenger RNA, which is the positive sense RNA strand, that can be translated for protein right away. All right. I think that's enough to chew on for today. Right? And we'll finish up with the rest of the information on Wednesday.
So when I was confused, you were talking about, just to clarify so I understand, right? You were talking about viruses infecting bacteria.
In the beginning. For the
That's where I got lost. I thought we were talking about peoples. Okay.
Yeah.
All right, thank you.